Looking for Grace. Oh, okay. So I thought there's just like a funny place to leave your water bottle. Okay, good enough. Okay, boy do I have bad news. We don't have class on Wednesday here. I know. Don't be emotionally overwrought. I realize you'll get through without your daily fix of me or every other daily fix of me. I'm not going to be back on campus till Friday. I will record one lecture for both sections and distribute that. I might even get it done tomorrow at some time. It's one of those days you got to wait at your house for somebody to show up, service person, for like 3 hours. We'll be here between 11 and 4. To be a little more specific, okay. But I'll be back, and an email will be fine. I just have to be. Me and my wife have to run down to DC for some family stuff, and we'll be back. Moving on. So without further ado, I'm going to finish up digestive today, and I have got time to at least introduce a little bit about the urinary tract. We'll go from there. So here's kind of where we were. I better pop that back in. I clip that one out. I will get there just a bit. I don't know where we should, but I'll just review what they were doing. And so I basically am kind of reviewing the small intestine because I think it's really important, starting there and finishing up from that point. We waited for you. Tell Grace she has to be here on Wednesday. All right, moving on. So the small intestine, the reason I want to emphasize it, this is where digestion takes place. It's all by this time, the mechanical stuff is done. Chyme is in there. It's just the rest of the chemical digestion, the absorption, and the magic happens inside of 8 feet. Really, the vast majority of everything is done. Again, there's 3 components. You have the duodenum, which is that little C-shaped part where it starts around the head of the pancreas. The common bile duct is there. That's why it's so vital. And then you have 8 feet of jejunum and about 12 feet of ileum. That's ILEUM. And by the time you get through that 8 feet, it's basically done. It's all identical. Other than the common bile duct, everything is the same. Okay. And it's that amazing, this 22-foot tube has a tennis court worth of surface area, and that's really the important part when we look at it anatomically. So it does all of these things pretty straightforwardly. Here are the portions of it. Yes, the mesenteries hold it in place. The omentum covers it anteriorly, so it's like a fatty layer of protection against the anterior wall that's there. It's a very well organized system, and thankfully, even though it's essential, we have a fair amount extra in case something bad happens, like there's an injury or an illness where a portion of it might have to be removed. There's plenty of like extra footage, so to speak. But the big thing here are what creates all the surface area and why it's so magical, and why we can basically chemically digest and absorb nearly everything in a matter of 6 to 8 feet of intestine. So you will see here all those illustrations sort of summarize it to a certain extent. It starts off with basically this is the first way I think to start it. And I'll show you the other illustration. Matter of fact, maybe do it backwardsly so you can see what I'm talking about illustratively. Hear you beeping. This thing out of my mouth. It's killing me. Never put a Werther's thingy in there because they take too long to dissolve. Oh, they're good. My wife and I are addicted to those. We never buy them because we'd be addictive. So here it is. These are, they call them circular folds or plica circularis. It's a term I use. You'll see that on your PowerPoint. Villi and microvilli. So this is the big player here. it's a little, these plica, or the way it's folded, are a little, rather than passing the chyme lengthwise like water would go through a tube, like a garden hose, it spins around. So you get maximum contact through all these little grooves that are about a centimeter deep. And on top of them, if you could magically put your finger on these little millimeter tall nubs, they're like little nubs, but in reality, they're finger-like, and they're covered completely in them for surface area. But it'd be like little bumps to you and I. And each of those tiny little bumps are like a finger that's covered completely in cells, simple columnar cells, that have increased surface area because the edges, the outer edge of the cell, are covered in microvilli, basically cellular extensions that increase surface area. It's all about surface area. So you get surface area that's bigger than this room out of a 20-foot tube that's about, maybe an inch in diameter. Imagine that. That's the beauty of this particular situation. So each of those little nubbies proportionally are like a finger, and in the middle of the finger is a lymphatic vessel that's there to absorb mostly fats and heavier fatty products. And it's a lymphatic capillary called a lacteal. And then it's surrounded by a regular capillary bed that absorbs whether it's amino acids, whether it's glucose, whether it's other small molecules like nucleotide-based molecules, things like that, easily absorbed through there. And here you can see their idea of the microvilli, the brush border that's there. And what's neat about that brush border are tons and tons of brush border enzymes, because effectively what the chemical digestion does is it breaks it down to nearly the smallest component. So you have little peptide chains that become amino acids. We suck those in. You have lactose and maltose and sucrose that we break down into monosaccharides, effectively glucose, for lack of a better way to express it, and it's sucked in there. And so understand that that's what those enzymes do. They're so important. The enzymes that are really in the center for chemical digestion are prepping it for those brush border enzymes to be the final factor that helps bring them in or absorb them. Because it's a size situation. Big things can't get through there. They have to be chemically adaptable to get through that boundary and be absorbed. If they're fatty based by the lacteal, unfortunately, bacteria, viruses, microorganisms can get in there. And the vast majority of the stuff gets into the blood vessels because they're really compatible with plasma. This is what it looks like microscopically on an actual micrograph. These are the brush border, and you can see all the mucus granules, which contain a lot of the enzymes that are in this. Again, surface area, surface area, surface area. So I did want to show you that, and I'll take you back to the other PowerPoint that we've been using. And so the circular folds are plica. Plica means fold. Circularis, okay, surface area. The villi, surface area. And that's where the absorption takes place. Each villus has the capillary and the lymphatic, the lacteal. Microvilli cover it. And you're going to see adjacent to it when they show you their illustration, nearly at the base of all of these fingers are tiny indentations. And where we have gastric pits in the stomach, we have intestinal crypts. I know it's confusing. They're basically the same thing. They're a gland. They just have a fancy name called crypts of Lieberkühn. And really what's interesting about them, that's where most of the activity is going on with regard to the chemicals that are being produced, where a vast amount of, a lot of the cholecystokinin, the secretin, a lot of other chemicals are produced. And those are the cells that are the most rapidly reproductive, effectively, let's say, in the mature body. When we're no longer at neonates or infants, when you might have other tissues, these are every 3 to 6 days. I beg your pardon. These are probably every 2 to 4 days where these cells are replaced. That is why that is the area that is most sensitive to radiation and chemotherapy when people are getting treated for cancer. And that's why that's the typical side effect of nausea, diarrhea, vomiting, gastrointestinal symptoms, and signs and symptoms are so rampant in people who have to undergo these treatments. It's really the problem. My colleague next door, Dr. Buxton, and I co-taught this class for 3 or 4 years in radiation biology for the folks who were in nuclear medicine. Now they have someone else who's able to teach it for us. But we did a good job helping to develop and get that course sort of good to go. Here you can see a better view of that lacteal. Here you can see the intestinal crypt. It's really just another gland, similar to the gastric glands. And again, all the secretions are there. Here you can begin to see them. Each of these individual areas that are there, the lacteal that are in them, little indentation. Here you can see it's a simple columnar cell interspersed with goblet cells. And this is a really good illustration of microvilli vis-a-vis surface area. It's great to see those types of things because I think they speak better as to how much surface area is developed. That's there, and even a little bit here. Again, here they're showing you the circular folds as it travels down. There used to be something at the Science Center where you could put a quarter in and watch it. I don't know if it's still there. I haven't been. My first wife worked part-time as a presenter in the Science Center. And that's how my son, who's in the Navy, got his start. He helped with people in the submarine museum. He actually, as an aside, his senior project in high school, he went and interviewed the captain during World War II, was the first captain of that ship. It's a boat, not a ship. At Annapolis, I had a conference down there. I dropped him off at Annapolis at the Naval Academy, and he went and found the instructor. The rest is history, as they say. It was also a great place to watch the fireworks on July 4th. Sitting on top of that. You don't like fireworks? This is Pittsburgh. This is fireworks central. Oh, God. You know, it's hard. I mean, we have Eagles fans. Okay, moving on. So mucus, watery fluid, okay, helps to transport. A lot of these enzymes, these are what we call the brush border enzymes. This is the part that helps to finally break it down to amino acids. This is the kind of, so when you hear about somebody who's lactose intolerant, they don't have this enzyme. Understand, I know, I'll point out later in the PowerPoint, I have family examples about that from two of my wife's sons who, as adults, older than you folks are now, became lactose intolerant, who never were when they were kids. Okay, one who developed celiac disease, gluten intolerant and lactose intolerant as a man over 30. Go figure. The body's very funny like that. The fact that you didn't have a problem doesn't mean you won't get a problem. Same with food allergies. They can come almost any time. Allergies are like that. So, I mean, it's interesting. And a lot of times I see it more from the gut related things, but it's in token they go away. My middle child was lactose intolerant as a kid. Now he doesn't have nearly the problem with that. I mean, he just has to be careful not to overdo it, but he doesn't have a big problem with it now, comparatively speaking. So, and again, the big chemicals that govern here, secretin and cholecystokinin, they're like the gastrin in the stomach, lots of antimicrobial substances, a good summary of it. We do not need to go into all the detail and know all the names that are there. I mean, I do have my advanced phys class. I mean, I don't want to subject you to the same criteria because it's not really necessary. I mean, for most of you, I'm trying to give you a generalized acquaintance. I think, and the charts like this are good because they give you an idea where things are, and maybe that's something you're going to have to know going forward. You can come back and look at these later. So again, stretch receptors. This is an alkaline area. pH is up. It neutralizes the acidity. And again, what stimulates it when food starts to enter by way of that, when you look at the end of that, the pyloric sphincter, where it begins to enter into the duodenum, that triggers everything. As food begins to come through, it stretches the areas that haven't been stretched, and then that's mechanical stimulation. Gastric juice is acidic, that's chemical irritation. And all of those types of things, the goblet cells make mucus, a lot of times it's bicarbonate rich, distension of the wall, that's stretching it, okay, stimulates parasympathetic reflex, that triggers the secretin and cholecystokinin. The system it works by itself. You don't have to give it a second thought. We don't need any overrides whatsoever. That's the beauty of a lot of our systems vis-a-vis homeostasis. It's fine. As long as nothing goes wrong, it is fine. It's there. I mean, it is. And to some extent, a lot of us kind of, you ignore it. You have to, down the road as you get older, you have to start looking for little signs. We'll talk about that in a second. They're there. Villi for surface area. The small intestine, it's not just most important, it's vital. We cannot live without it. To be clear. Very little absorbable material. These are the kinds of things. Carbs, we haven't touched carbohydrates since saliva. The pancreatic amylase enters in the small intestine, breaks it down. Principally and effectively, we're absorbing monosaccharides. I'm not going into the chemistry or the mechanics of how things are brought in and absorption. There's a reason for that. I'll just summarize it. Again, it relies a lot of this movement, and the same is true in the kidney when we get to the kidney. Bringing stuff in, okay? We utilize sodium a lot. We have so much sodium that you might remember way back in AP1, we talked about something called primary active transport. Basically, we bring in sodium actively, and because there's so much, we create flow patterns. So we can bring things along with the flow, that's called co-transport. As we bring things in, we create differences so we can push things out using the same kind of energy. It's almost like backwash. We bring it in, it bounces off, and it comes back out. That's called antiport. We utilize that a lot here, but sodium is sort of the linchpin. Why it plays such an important role of how we get it into those cells and how we get it out of those cells and into the bloodstream. So again, this is the type of stuff I just did with my advanced phys class. I'm not doing it with you because I'm just trying to give you the quick summary about how it works. Proteins, they have to be, we break them down into peptide chains, polypeptides, finally smaller and smaller until we get amino acids. Those we bring in. And that's the note to understand. Everybody, maybe you do it yourself, people who want to work out, want to get, want to get built up, they ingest protein powders and muscle milk. That's all fine. Two things. One, drink a lot of water, because your kidneys have to get rid of the excess nitrogen. We'll get to that with the kidney. Number two, okay, it doesn't, it's not like magically your muscle milk, whatever it is, creatine, it doesn't magically make itself into muscle. We absorb amino acids. Effectively, those are building blocks. How we use them depends on everything else. If you're exercising, if you're on a plan, if you're eating other stuff responsibly that assists the process with a training program, it's going to build muscle. I mean, if I do it, it's not going to build muscle because I'm not in that kind of a plan to do it. And as you get older, you're probably not wise to do it because your kidneys can't probably process it as quickly as they should. So understand, it's like people have this notion, oh, it'll magically, I drink this these proteins are what muscle are made of, it'll immediately make muscle. It doesn't work that way. So people are a little silly about that. This, again, is just some of the stuff we looked at AP1. Fats, by and large, if they can make them small enough, the glycerol, remember triglycerides, that can be absorbed fairly easily into the bloodstream. It's the fatty acids. If they're just individual fatty acids, they're small enough. When we get these complexes, depending on what you've ingested, we have to create different things that will allow these fats to be carried in. And a lot of times, the way, if they're not in the lacteal, they're transported by something, okay, called chylomicrons. And the chylomicrons are a problem. Even if they go in the lacteals, they're still going to be in the bloodstream. If you eat an unusually fatty meal, you end up with a lot of this in your bloodstream rather quickly. And if you have pre-existing problems with blood vessels or a tendency, hereditary tendency to get arteriosclerotic heart disease, some type of vascular disease, you have to be very, very careful. I've made this point. I'll make the same point with you, just additively. If you can, try to make the fats that you eat, as the vast preponderance of them, healthy fats. Okay, healthy fats are omega-3s, not omega-6s. Omega-6s are pro-inflammatory. They're nasty for your circulation. It's the difference. I mean, there was a big article I was reading about different oils that are so. Everybody, they fall in love with vegetable oil and canola oil. They're cheap for a reason. Okay, they're inexpensive. Are they useful? We call them neutral oils because they don't have any taste to them. They are rich in, unfortunately, omega-6s, which are not healthy for you. I mean, the two best oils to use in no particular order: good old EVOO, extra virgin olive oil, and avocado oil. Those are the only two oils we keep in the house. Not cheap. That's the problem. Those are omega-3 oils. Oh, we don't leave home without our EVOO or avocado. Love that. I mean, you should have been there for the mango shrimp. Oh, to die for. Thai recipe, delicious. Okay, moving on. You don't have to do that. I mean, this is the pattern of absorption and how they go through and into the lacteal, and don't worry about that. You can look at that. Not a big deal. Yeah, the mechanical activity is motion more than anything else. And there's some peristalsis and there's a fair amount of segmentation, because what happens is optimizes that segmentation, that back and forth, thoroughly allows it to be absorbed and processed, as opposed to the peristalsis. So the peristalsis will keep it moving, so to speak, but this is maybe the primary activity that's there. Again, parasympathetic impulses. This is one of the things why somebody, if they're lactose intolerant and they ingest something with a milk load, all of a sudden, boom, it's almost like they're racing to the bathroom. It triggers what they call a peristaltic rush. It's really a problem. It surprises you how quickly that occurs. It's almost like a reflex activity. Be the best way to describe it. So don't minimize that type of a thing. That's about all for small intestine. Large intestine, not a lot happens. It's non-essential. You could do without it. Obviously, you would like to have it. It's only about 5 feet or so long. It's maybe about, I'm going to tell you, maybe 2.5 inches. It's got very distinct anatomy. It enters at something called the ileocecal valve. That's the stricture that occurs there, the narrowing or the valve. Cecum is the first one of these components that's there. And really, what it does? Water, it utilizes water to help what's going to become feces move. Okay, eventually absorbs it till it gets the right consistency. I hate to be gross, not too dry, not too wet. Maybe some electrolytes, maybe some vitamins. That's about it. And all that stuff we can deal with elsewhere. That's why it's not essential. It's there. And really, I'm going to go forward just real quickly. Here it is. This is the valve, the ileum and the cecum. That's there. Each of these sort of enlargements, the singular is haustrum. We don't see that in the small intestine. Here you see these pouch-like formations. So collective, more than one haustra is a plural. The first haustrum has its own name. It's called the cecum. It's the largest of these containers. And jutting off of it, as you can see there, is an opening into this structure, which is a lymphatic structure called the appendix, also known as the vermiform, Bermiform, worm-like appendix. Its job is immunological in nature. Yes, it's collecting bacteria, but really what it does, most authors would agree, is when we have really bad diarrhea, we lose a lot of the natural gut bacteria that are so important for vitamins and electrolytes and really things that keep us healthy. And so it reseeds it. Now, years ago, I mean, they would take out appendix like crazy. Appendices, the appropriate plural. But nowadays, the inclination is to leave them in because they're immunized. It's like tonsils and adenoids. If they're causing a problem, they have to go. If they're not, try to keep them. It's really the approach that's there. The other thing that's notable anatomically, as well as the haustra, is this. There's three of them. They're bands of smooth, and they keep that shape, that typical shape of the ascending colon, the flexure. It's only tacked down here, retroperitoneal, on the hepatic and splenic flexure. It's the transverse, it's the descending colon. It maintains the shape that's so appropriate that's there. And then you have these little fatty, they look like raindrops, like droplet type shape. They're called epiploic appendages. What they do, nobody really understands. But those are the three hallmarks anatomically: haustra, epiploic appendage, and the tinea coli, or tendon of the colon. Because anatomists thought it was like a tendon, but really it's smooth muscle. It's there. Everything else nothing really remarkable to report. The cecum is the start. Ascending, descending, transverse. As you'll see, as it gets down there, the sigmoid, because it has an S curve, and that sort of takes it from the outside and sort of brings it in centrally, kind of goes down and sort of ducks inward, if you will. And so now it's central, and then as it turns around, now you develop the rectum, which leads into the anal canal. That's the transition point between the simple columnar and finally stratified squamous non-keratinizing, because now the feces are formed and they're abrasive. They're going to denude more cells as it goes down. Stratified epithelia are for protection. And you can see at the last, couple inches, opening into the anus outside. There's sphincters, internal, we don't control, external, we do control. Okay, basically on guard. Here, and again, this is a lot. There's a lot of blood vessels. There's a lot of musculature associated. It has this column-like appearance internally. What's interesting is this is an area where you do see a lot of hemorrhoids, enlargements of these veins. You see a lot, various conditions, but pregnancy, particularly when it rears its head. Other people for different reasons. A lot of times it's diet. Same layers, everything else the same, et cetera, not much here. Effectively minimal digestive. These are basically almost like a lubricating mucus. If there's any water comes in, most of it is absorbed on the way out again to finalize the fecal content, intestinal flora. So they do break down cellulose. That's where gas comes. So some people have more problems than others. It's probably more diet related. We all produce a certain small amount of it. You can't help it. The vitamin K, B12, and thiamine, all of those can be absorbed there, but they're also absorbed elsewhere. You should remember, not for me, but for future, thiamine is B1, riboflavin is B2, ascorbic acid, vitamin C. Some of those names, particularly if you're taking licensure exams, you see those again. They won't just say thiamine. They'll say, thiamine is blank. You should know B1, riboflavin is B2, ascorbic acid is vitamin C. Know your names. And not a whole lot. Again, there's a lot of reflexes that are involved. I don't think it's a big deal. Again, the vast majority of feces is water. The rest is undigested food and cells. It's about a third. I told them the same anecdote I will tell you. So after my first major stomach surgery, I said, when can you get this stupid tube out of my nose and I can eat something? Because I was limited to the occasional icy chip for 7 days. When you finally go to the bathroom, why is that? The doctor, stupid question. You're not giving me anything to eat. He goes, you know this, doc. 1/3 is water, 1/3 is cells, 1/3 is undigested food. You still got 2/3. You're going to go. Eventually, you go. It does work that way. Shocks people, but it's absolutely true. They wouldn't feed you because they want to make sure it's working effectively. You're sort of at cross purposes. Oh, it's very frustrating. Having gone through that on 2 separate occasions, if you ever have a nasal gastric tube, you don't want it in there long. It's not fun. So, things that happen, you won't have to worry about this. As we get older, nearly all of us have problems with this. They're called diverticuli. I just had a colonoscopy in September, routine. Incidental finding, yeah, there's evidence of a diverticulum. It's like an outpouching. The smooth muscle wall weakens. You get like a bubble in a tire, an outpouching of the material that comes out and protrudes. Stuff can get trapped in there. It can get infected. It can irritate. It could even burst. And so diverticulosis is the name of that. The inflamed and infected is really a problem. The inflammatory bowel disease you wouldn't want to wish on anybody. Most of the time it's autoimmune in nature. Certainly this is a very significant cancer. It's easily testable. Those color guard, those commercials you see littered. Basically, once you reach about 40, these are the kinds of things your physician will tell you to do or you can do on your own. Basically, give them a fecal sample, you take a small amount of it, mix it in some kind of solvent that they have, and basically send it to the lab, and they tell you whether there's something bad in it. Mostly they're telling you if there's blood in there and anything else. And if there is, then you need to get it appropriately tested. Typically, the interval, once you're past age, 40 or 50, whatever the rules are, it's about every 5 to 8 years. You need to have one of those done. So, like I said, I had been a good more than 8 years without an endoscopy or a colonoscopy, or somebody who's already had major GI surgery on 2 occasions, albeit the last 25 years ago, but I had them both done in September. And in January, when we started here, I had a quick follow-up because that's where the surgery was, just to make sure that where the surgery was, there was a tiny polyp that was not a problem. And it wasn't. So I got a good report on that. And all these things. You know what this means? Don't get old. And my wife is a bright woman. She made one mistake. She married some old guy. So don't marry somebody who's, nearly 7 years older than you are. If you're living with an old guy. I digress. All right, introduction to kidney. This is good. We're in the same point where the other group was. I'm happy with that. Well, hello, meet the kidney. I didn't load your, I'll get that done between now and when I see you again. And, I'll see what they're covering for this. This is the Marib one. This is what we're talking about. I know you had the anatomy recently with my colleague, Dr. Robbins. Understand. So I try to focus on the physiology a lot. You have to understand the anatomy to understand the physiology. I am not going to go crazy. The physiology is very complicated. To start. See, okay. We'll start with this first. Job 1. Yes, the major excretory organ maintains the body's internal environment by job 1, metabolic wastes. Nitrogenous waste removal. We have all those wonderful proteins and we have an abundant culture here, lots of food, wealthy societies, people tend to eat a lot of proteins. The trouble with eating proteins are the amino acids. I just talked about them. When we get an amino acid, we have to remove the amino group. You don't have to know the chemistry. That amino group, when it's removed and gets into water, it forms ammonia, NH3. Ammonia is a horrible toxin. The liver magically takes that ammonia, takes two of those ammonias, and carbon dioxide. How's that? Something that we a waste product that we can use in our bloodstream, not just to make carbonic acid, but we can take two of those, mix it with the carbon dioxide, and voila, we end up with that C double bond O and two NH2 groups. And it's called urea. It's 100 times less toxic. That's what your liver does. It detoxifies. This is its major detoxification. How does it do it? It makes things more water soluble. So it makes it easy for the kidney to get rid of. We have two major nitrogenous waste products. From proteins, okay, we get urea. From nucleotides, from DNA and RNA, all those cells were building up and breaking down. We can't recycle everything. There's a lot of DNA, RNA. If you remember purines, remember DNA are A's and G's, T's and C's and U. The A's and the G's are called purines. Their waste product is called uric acid. Those are two nitrogenous wastes we have a limited tolerance for in our bloodstream. That's the big job of the kidney. And on top of that, the kidney has to regulate water and regulate what's dissolved in the water. So all those ion concentrations and all the fluid outside the cell, and they impact what's inside the cell. Acid-base balance, because that's ions, hydrogen plus, HCO3 minuses. So all of those things, this is the only mistake. They've got these backwards. Renin, we've covered a couple of times, we're going to cover it yet again. That's that renin-angiotensin aldosterone mechanism. That regulates blood pressure. Erythropoietin regulates red blood cell production. We went over that. It stimulates the bone marrow to make red cells. So they got it. And that's what I mean. PowerPoints have mistakes. So every time I see one, I try to point it out to you. And they do all the time because they're not subject to editorial review. The kidney is so important and so hard to understand. Bless you. Very, very difficult. So, I mean, I'm not going to go into all that stuff, a little bit of it. Not done yet. Vitamin D, which we have to have for basically to absorb a sufficient amount of calcium through our diet. We're a principal source where we get it. And if necessary, if we need in an emergency like fight or flight, this is another area that can make more glucose. That's what gluconeogenesis, you don't have to know that. Years ago, we used to talk about the upper tract and the lower tract. We don't do that. We have the kidney. Everything gets done in the kidney. That's the function. The urinary tract is basically the urine passing out. So there's no processing that goes on. The ureters are tubes that take it from the kidney. It's temporarily stored in the bladder, and it ends up being excreted by way of the urethra, whether you're a man or a woman. Only bad things can happen if those things don't work. Kidney stones, inflammation, sexually transmitted or non-sexually transmitted infection. And if you're an idiot who smoked for 40 years, 2 packs a day, bladder cancer. So inevitably there'll be a question about that. Just saying. I'm happily here to tell you about it. I told the other, you guys must be sick. With me not being here Wednesday, that basically leaves you and I having personal encounters for maybe at tops 4 more, maybe 5 more times. You're happy to hear that. Tired of hearing about his family, no more stupid anecdotes, nobody cares, stop with the bad jokes. I know what you're thinking. AFT, about time. There they are. Okay, basic anatomy, they're lower. There's a reason they're lower. Liver sits on top of it. Okay, this is the whole ball game right there. Notice the blood. I talked about this a little bit in the anatomy. Okay, and maybe it was emphasized. This is called the inferior phrenic. This is the adrenal or suprarenal artery. Look at the size of the renal artery. Look at how small the inferior mesenteric is. Superior mesenteric. Look at how wide that is proportionally. It's all about the anatomy. The kidneys are a high pressure filter. They only work if you have sufficient blood pressure. When your blood pressure drops for whatever reason, all the different things we talked about with shock, your kidneys have one response, they shut down. Your kidneys get an infection, they shut down. Your kidneys get a kidney stone, they can shut down. Okay. The kidneys have one response to adversity, they shut down. So this is the big problem, and that's what happens with shock, and it's devastating. Everybody here, understand, I ended up in ICU because of that, from an infection. That's because I ended up with an infection that caused my blood pressure to drop precipitously, and I ended up in ICU for 3 days. Nothing. Nearly died. Having nothing to do with the aforementioned stomach surgeries. So it's about that. So that doesn't really help you. And this doesn't really help you. Okay, the blood vessels and the ureters enter and exit at someplace called the hilum. The liver has one of those. Not a big deal. Yeah, they're protected by the ribs. The bottom of the kidneys are a problem. I mean, they're not as well protected, and so you've got to watch out for trauma. When someone's in an auto accident, we always routinely, they come in, and your significant abdominal or back or side trauma, you start worrying about. That's why we catheterize it. We try to get a urine if they're out of it, and you're just trying to keep them going. You want to take a look at, you catheterize them to see if there's blood in the urine. If there is, you have to work it up. Okay, not a big deal. There's a little bit of fat around them. The interior is very membrane-y, kind of like the lung that you did in lab. It's got blood vessels and membranes and tubes that are soft. The exterior, the capsule, is quite strong, quite sturdy. It's there, and there's a little bit of fat around it. This is what's meant by retroperitoneal. I mean, the organs are all in here. They're moving around. These are stable. Important things or important places always are held stably. And they talk to you about this. All of these are types of things that you worry about it. Okay, car crashes. I mean, big problems. We look for that sign, and there's different degrees about that there. Effectively, what's neat about the kidney, you have an outside and the inside. The cortex is the outside. That's where all the filtration occurs. The medulla is all about concentrating the urine. I'll show it to you. So this is the anatomy. Easier to show it this way. The blood vessels enter here, and they exit here. They go up. These are the pyramids. Base is this way, the tip is here. Effectively, urine drips out of these tips. These are called calyces, small ones that lead into big ones. At this point, it's urine. Nothing else happens to it. Processing is complete as it comes out of here. And effectively, it enters a series of funnels, the largest of which is called the renal pelvis, the stem of which is the ureter. In the meantime, the blood entering here is cleansed. Ridiculously, about 30 times a day, your blood runs through completely through all of your blood runs through your kidneys. Okay, it's remarkable. And what happens is that once it's cleansed, it comes back out by way of the veins. These are called columns. In between the pyramids are renal columns. That's where the blood vessels travel. So I will show it to you here. Effectively, they come in this way through these columns, and the veins go out. By the time they get into the cortical area, they begin to arch over the pyramids. They're called arcuate or arching arteries. And then they begin to radiate. Little spikes come off of them, and they form glomeruli. Glomeruli are capillary beds, and they are the foundation that we're going to get to next time of the nephron, the whole reason the kidney works. All the filtration happens here. The vast majority of reabsorbing or getting rid of, bringing back stuff we need, getting rid of stuff we don't need occurs here. And in these pyramids, it's all about concentrating the urine because we're reclaiming, are you ready? 99% of the fluid. You're going to see we generate about 160 to 180 liters of fluid. 99% we reclaim. Only 1.5, typically 1.5 to 2 liters of it is excreted a day, what you and I call urine. And this is the important part, and the last part I have to tell you about today. Nor is the only place I know of in the body where you have an arteriole oxygenated blood going through a capillary bed and ending in oxygenated blood. Why? Because pressure is what's creating that filtrate. We use the pressure, the high pressure, that wide renal artery, and the high pressure. It's like putting water into your coffee, into a drip coffee pot. We're using gravity, okay? Water pressure, it's going to go through. The same thing here. We're using blood pressure to create that filtrate so we don't have to expend any of the energy, utilize any of the oxygen. Then, when we want to either concentrate by way of what's called a vasorecta, or we want to reclaim or secrete, reabsorb or secrete electrolytes, glucose, chemicals, all the good stuff, that's when we expend the energy. So it's a really unique system where an arteriole enters a capillary bed and exits as an arteriole, preserving that oxygen, preserving those nutrients to be able to do the rest of the work. It's a fascinating system. I'll leave you with that. I'll probably send you a dictation starting with talking about these nephrons and how they're unique and what they do and how they're constructed next time. Thank you.